Hello, my name is Mike Princiotta, and I am the CSO at EpiVax Therapeutics. I'd like to thank the organizers for the opportunity to speak at the ISV Annual Congress. I'll be talking today about the efficient selection of tumor-associated neoantigens for the use of the design of personalized neoantigen vaccines. I would like to start by introducing EpiVax Therapeutics. EpiVax Therapeutics began life as EpiVax Oncology, a spin-off from EpiVax Incorporated. EpiVax is a privately held company founded in 1988 by Annie DeGroot and Bill Martin, who developed the EpiMatrix and Janus Matrix computational tools used to predict MHC Class I and Class II epitopes in infectious disease and biologics. And their technology is the basis for the discovery of the T-regitope concept. EpiVax Oncology was founded in 2017 with the express purpose of leveraging the EpiVax Bioinformatics platform for the development of a therapeutic personalized neoantigen cancer vaccine from preclinical design to phase 1 clinical trials. In recent months, we've expanded the mission of what is now EpiVax Therapeutics to include the development of a T-cell directed COVID-19 vaccine. However, I will keep today's presentation focused on our neoantigen cancer vaccine program. While cancer immunotherapy has been around for some time, it is only recently that significant advances impacting long-term survival of cancer patients have been seen in the clinic. Most of these advances have been centered around T-cell-based therapies, especially the use of CAR T-cells for the treatment of hematological malignancies and antibody-based checkpoint inhibitor therapies targeting CTLA-4, PD-1, and PD-L1, which are expressed on the surface of effector T-cells and limit the effectiveness of the anti-tumor T-cell response. The idea behind therapeutic neoepitope cancer vaccines is to improve on the efficacy of checkpoint inhibitor-based monotherapies by stimulating more robust T-cell responses against peptide antigens that are unique to a patient's tumor. Precision cancer immunotherapy in the form of personalized neoepitope cancer vaccines is possible because of the advent of rapid next-generation sequencing. The ability to sequence a patient's tumor and healthy normal tissue allows for the identification of non-synonymous mutations that may be seen as foreign by the patient's cellular immune system. Since these mutations will be expressed only by tumor cells, T-cells will target the tumor cells for destruction, but not healthy tissue elsewhere in the body. The challenge to designing vaccines that will direct T-cell responses to these tumor-associated neoepitopes is in identifying which peptides expressed by tumor cells and containing non-synonymous mutations will be immunogenic. It is important to note that one of the challenges faced with more traditional cancer vaccines has been the need to generate T-cell responses to non-mutated peptide epitopes, as occurs when targeting tumor-associated antigens, or TAAs as they are commonly referred to. Even if the tumor-associated antigen from which a peptide epitope is derived is expressed exclusively by tumor tissue, the peptide does not contain a mutation and will be seen as self by the cellular immune system. High-affinity T-cells recognizing self-antigens will have been deleted during thymic selection. As such, it is difficult to generate effector T-cell responses against epitopes that are either self or have high degrees of homology to self-peptides. To this end, EpiVax Therapeutics has developed a predictive algorithm called Answer that utilizes the EpiVax, EpiMatrix, and Janus Matrix bioinformatics platforms. Answer is designed to select tumor-associated neoepitopes that are most likely to be seen as non-self by the host's cellular immune system and will, therefore, be more immunogenic than neoepitopes that are more highly homologous to self-peptides. Identification of tumor neoepitopes using Answer follows what has become a relatively well-established and accepted protocol for the production of personalized cancer vaccines. A tumor sample is collected, typically through some form of biopsy, along with a sample of normal tissue, which is often a peripheral blood sample. Tumor and normal tissue are analyzed by whole exome sequence, and variants in the tumor exome sequence are called by comparing to the patient's normal tissue exome. A patient's HLA type is determined either serologically or from the exome sequence data. The EpiMatrix algorithm is then used to scan the variant call files to identify neoepitopes specific to the patient's tumor. These neoepitope sequences are then analyzed using the Janus matrix algorithm, which identifies and removes self-like sequences from the list of neoepitopes generated using EpiMatrix. By identifying and removing self-like peptide sequences, both regulatory Tcell and potentially cross-reactive epitopes will be eliminated from the final neoepitope output generated using ANSWER. The final step is the ranking and selection of non-self neoepitopes. These sequences are used to design short polypeptides that contain HLA class 1 and class 2 neoepitopes that can be then manufactured for use in a personalized cancer vaccine. ANSWER is designed to optimize the design of polypeptides for inclusion in a vaccine formulation. These polypeptides will often include multiple overlapping neoepitope sequences that can be presented by both the patient's HLA class 1 and class 2 molecules. The entire process is locked and hands-free, ensuring reproducibility of results and aligning with regulatory requirements for bioinformatic neoepitope identification. The process is rapid and designed to have vaccine-ready sequences from raw exome sequencing data in no more than 2 to 3 days. Conceptually, targeting tumor-associated neoepitopes to control tumor growth is relatively straightforward. However, translating this approach into an effective immunotherapy is a bit more challenging. Most algorithms designed to identify peptides that will be presented by HLA molecules rely primarily on peptide HLA binding affinities for selection. However, this is only half of the equation. Peptide epitopes also have an outward-facing surface that is recognized by the T-cell receptor expressed by the T-cell that will respond to the HLA-presented peptide. Janus matrix is designed to predict the cross-conservation of epitope clusters in the human proteome based on these T-cell receptor-facing peptide sequences. By comparing neoepitope sequences for conservation with self-peptides, Janus matrix is able to identify and remove in silico inhibitory or tolerated epitopes. The identification and removal of these self-like peptide sequences in designing personalized cancer vaccines is important with respect to generating an effective vaccine. Not all peptide epitopes elicit the same response when presented to T cells by HLA class 1 or class 2 molecules. Non-self-like sequences are more likely to be recognized as foreign and therefore elicit effector T cell responses, whereas self-like peptides are more likely to generate inhibitory responses and, potentially, responses that will cross-react with self-peptides expressed by healthy tissue. It is important to note here that ANSWER is designed to select peptides presented by both HLA class 1 and HLA class 2 molecules. Most algorithms focus on HLA class 1 presented peptides. Regulatory T cells are primarily CD4 positive and recognize peptides presented by HLA class 2 molecules. Without the ability to predict HLA class 2 epitopes, it is not possible to identify and remove neoepitopes that will produce an inhibitory regulatory T cell response. Regulatory T cell epitopes can potentially be found in any protein sequence, and it is important that they be identified and removed from a vaccine formulation. A 2014 publication from Losikoff et al. Describes the identification of a regulatory T cell epitope from hepatitis C virus that is homologous to multiple human protein sequences. Importantly, this epitope is able to induce a regulatory T cell response in infected individuals. To illustrate how Janus matrix is used to remove potential regulatory T cell epitopes, we show here the identification of two distinct epitopes from the tumor-associated antigen Milan-A. Peptide 1 has a sequence that has a high degree of homology to multiple human protein sequences, similar to the hepatitis C virus peptide, and this is reflected in the high Janus matrix score. Peptide 2 has a low frequency of homology with known human protein sequences, which is reflected by the low Janus matrix score. By selecting peptides with low Janus matrix scores, we reduce the likelihood that a peptide will induce a regulatory T-cell response upon vaccination. To evaluate neoepitope selection using the Answer Bioinformatic platform, we used the mouse CT26 colon carcinoma tumor model. The first step was to perform whole exome sequencing on the CT26 tumor, as well as normal tissue from the Banalpsy mouse strain from which the CT26 tumor cell line is derived. Normal and tumor cell lines were compared, and a total of 378 single nucleotide variants were identified after quality control and filtering of the data. Using Answer, a total of 135 effector T-cell neoantigens were identified from the 378 single nucleotide variants. Importantly, neoepitopes with the potential to elicit inhibitory T-cell responses were identified and removed at this step. These neoantigens were then ranked, and the 20 neoantigens with the highest answer score were selected for evaluation. Answer selects for both MHC Class I and Class II presented peptides. Peptides containing the optimal sequence for vaccine delivery for each of the 20 neoantigens were designed in silico using Answer, and the sizes of these peptides ranged from 11 to 18 amino acids in length. It is important to note here that only 2 neoantigen peptides overlapped with those reported as immunogenic for the CT26 tumor model in the study from Kreider et al. That was published in 2015. Vaccines were then designed to deliver the selected neoantigen sequences as synthetic peptides or encoded using an RNA replicon or DNA expression system. We first evaluated the immunogenicity of the selected CT26 neoantigens by immunizing mice with the 20 neoantigens as peptides with poly-ICLC as adjuvant. Non-tumor-bearing Banalpsy mice were immunized 3 times at 2-week intervals, and spleens were harvested 1 week after the final immunization. Responses were measured by interferon gamma ELISpot against a pool of the 20 neoantigen peptides, and a very robust T-cell response is clearly demonstrated. One of the distinguishing features of the Answer platform is the ability to identify MHC Class II presented peptides that will be recognized by CD4 positive T-cells. As can be seen, a significant proportion of the responding T-cells are CD4 positive. Importantly, both responding CD4 positive and CD8 positive T-cell populations have multifunctional phenotypes as determined by the expression, in addition to interferon gamma, of TNF-alpha and/or IL-2. Expression of multiple cytokines is generally considered to correlate with greater T-cell effector function. To evaluate the breadth of the response to the neoantigens included in the vaccine formulation, we performed a deconvolution experiment where splenocytes from individual mice were tested against individual vaccine peptides. The immunogenic responses to individual peptides from 2 mice tested are shown in green. As can be seen, up to 65% of the vaccine peptides were immunogenic, which is a significantly higher rate than has been reported in the literature for this tumor model. What may contribute to this high rate of immunogenic neoepitope identification is the unique ability of the Answer platform to identify both CD8 positive and CD4 positive T-cell epitopes, as well as to filter out self-like sequences. Breadth of response is important in vaccine-directed immunotherapies as it decreases the chances a tumor will escape control by T-cells due to immune editing. In addition, a greater number of T-cell clones responding to tumor-specific antigens increases the opportunities for synergy with checkpoint inhibitor-based therapies. To evaluate how the inclusion of inhibitory neoepitopes identified using Answer affects the effector T-cell response to immunogenic neoantigens, a parallel experiment was performed where putative regulatory T-cell epitopes were included in the original neoantigen vaccine formulation. This experiment followed the same immunization protocol as just described, where BALB/c mice were immunized with 20 neoantigen peptides and poly-ICLC 3 times at 2-week intervals, and splenocytes were harvested 1 week after final immunization. However, an additional vaccine cohort was included where CT26 neoantigen peptides identified by answer as containing self-like epitopes and with the potential to elicit regulatory T-cell responses was added to the original immunogenic 20-peptide neoantigen vaccine. When these putative regulatory T-cell epitopes were included in the vaccine formulation, there was an 80% reduction in the effector T-cell response as measured by interferon gamma ELISpot assay. While the 20 neoantigen vaccine included both CD4 positive and CD8 positive T-cell epitopes, the inhibitory epitopes were all predicted to be presented by MHC class II molecules and, therefore, to elicit CD4 positive T-cell responses. This experiment clearly demonstrates the importance of filtering out potential inhibitory neoantigens. Being able to do this using a predictive algorithm without the need for in vitro validation makes this a powerful tool for use in the clinical setting. To test the effectiveness of vaccine formulations based on answer-identified neoepitopes, we performed several in vivo experiments using the CT26 tumor model. In the first experiment, BALB/c mice were immunized with the 20 immunogenic peptide vaccine formulation and poly-ICLC at 3, 9, and 15 days post-tumor implantation. A 30% reduction in tumor growth was achieved with peptide plus adjuvant treatment. A group of mice were treated with an anti-PD-1 monoclonal antibody after tumor implantation, as we wanted to assess the effect of checkpoint inhibitor treatment on vaccine-elicited tumor control. However, as can be seen here, the CT26 tumor line used at Charles River Labs is refractory to treatment with an anti-PD-1 antibody, and it was not possible to evaluate the combination of answer peptide vaccine with checkpoint inhibitor treatment. A second experiment was performed to demonstrate the importance of including an adjuvant when using a peptide-based vaccine formulation. In this experiment, immunizations were performed at 5, 8, 12, 15, and 18 days following tumor implantation. Treatment with adjuvant alone had no significant effect on tumor growth compared to PBS alone. When peptide and adjuvant were co-administered, there was a 48% reduction in tumor growth. It is interesting to note that vaccination with peptide alone actually resulted in a slight but measurable increase in tumor growth over vehicle control. While the mechanism for this increase cannot be determined from this experiment, it is possible that the lack of inflammatory signal provided by the poly-ICLC adjuvant results in a low level of T-cell tolerance to the peptide neoantigens in the vaccine. To examine the efficacy of delivering neoantigen peptides using a nucleic acid-based delivery platform, 2 experiments were performed. The first was in collaboration with Immunomic Therapeutics using their proprietary DNA plasmid technology. In this experiment, the immunogenic neoantigens were encoded in a beads-on-a-string approach, where the immunogenic neoantigen sequences are expressed sequentially using a DNA expression plasmid. This plasmid was used to immunize tumor-bearing mice at days 6, 14, and 21 following tumor implantation. As can be seen, there was a 46% reduction in tumor growth relative to mice immunized with the control DNA vector. This result is very similar to that seen with peptide and poly-ICLC in the previous experiment. In collaboration with Synthetic Genomics, a self-replicating RNA or replicon was used to deliver the immunogenic neoepitopes in the same CT26 tumor model. Neoepitopes were also encoded in a beads-on-a-string fashion in the RNA replicon. BALB/c mice were immunized at day 3 and 17 following tumor implantation. There was a 47% reduction of tumor growth relative to control animals, which is similar to that seen in mice immunized with answer-selected neoepitopes using either a peptide plus adjuvant vaccine or DNA-encoded neoantigens. A CT26 tumor cell line used at Synthetic Genomics is responsive to anti-PD-1 treatment. Monotherapy with anti-PD-1 in this model results in a level of tumor control of approximately 55 to 60%. When anti-PD-1 therapy is combined with the neoepitope replicon vaccine, control of tumor growth increases to 76% relative to control animals, demonstrating the benefit of combining vaccination with checkpoint inhibitor treatment. The T-cell response to neoantigen vaccination was further examined in the RNA replicon system. Here, tumor-infiltrating lymphocytes, or TILs, were harvested from tumor-bearing mice following vaccination. The frequency and cytokine profile of CD8 positive and CD4 positive T cells in the tumor were measured in mice receiving the answer vaccine, with and without checkpoint inhibitor combination therapy. A significant increase in CD8 positive T cells relative to control animals is clearly demonstrated in vaccinated mice, with the majority of T cells expressing interferon gamma only. When combined with anti-PD-1 treatment, the frequency of CD8 positive TILs increases, with the majority of this increase accounted for by T cells expressing both interferon gamma and TNF alpha. While there is no significant increase in the frequency of CD4 positive TILs following immunization, these CD4 positive T cells are largely polyfunctional, as measured by the expression of interferon gamma, TNF alpha, and IL-2. However, when combined with anti-PD-1 treatment, there is a roughly threefold increase in CD4 positive TILs, with the majority having a polyfunctional phenotype. Collectively, these tumor control experiments demonstrate a broad, multifunctional T cell response involving both CD8 positive and CD4 positive T cells to immunization with answer-selected neoepitopes. In addition, this response appears to be consistent across different immunization platforms and benefits from combination with checkpoint inhibitor therapy, at least in the RNA replicon expression system. One of the major challenges to the development of personalized cancer vaccines is the time needed to sequence the tumor in normal tissue, analyze the sequence data, and produce a vaccine. It is imperative that this process be accomplished in a clinically relevant time frame. Our goal is to leverage the peptide with adjuvant approach using the answer platform for neoepitope identification to facilitate the rapid turnaround times needed to allow earlier treatment options for cancer patients. We are doing this by reducing the time needed to go from tumor biopsy to whole exome sequence and integrating the bioinformatic steps that provide the neoepitope sequences used for vaccine formulation. Peptide synthesis times are targeted at less than 2 weeks for GMP peptide production, and drug substance and drug product formulation are targeted at less than 1 week. The goal is a needle-to-needle process that is no greater than 4 weeks. Rapid turnaround times for vaccine production would allow for treatment of patients at earlier stages of disease, where vaccines may have greater efficacy. As one example, by producing a vaccine in 4 weeks from the time of biopsy, it would be possible to treat patients in the neoadjuvant setting prior to tumor resection. Cancer vaccines may hold their greatest promise in the treatment of earlier stage disease, where they can prevent establishment of metastasis and recurrence of the primary tumor. Our goal at EpiVax Therapeutics is to make this possible. I would like to thank everyone for their attention, and I would be happy to take questions.